I'd like to welcome everyone to our third edition of the Journey to El Yunque webinar series. This month we have Dr. Grizelle Gonzalez, who will be talking to us about research related to the carbon cycle in the El Yunque rainforest and Puerto Rico more generally. Before we launch into the presentation, I wanted to talk a little bit about the Journey to El Yunque program. Since we will be focusing on the carbon cycling today, I wanted to highlight some of the aspects of the program for how Grizelle's research actually fits into what the students would do in the Journey to El Yunque program. So the main research question that students are investigating throughout the whole unit is that organisms in El Yunque seem to be well adapted to hurricanes, but what will happen to the ecosystem if the severe hurricanes strike the rainforest more frequently? So one of the aspects of the ecosystem is actually the abiotic resources and how the hurricanes affect the various resources in the rainforest, as well as how the changing of the biotic part of the environment actually also affects the resources. So we actually have a whole section of the program that deals with resources, and we try to help the students get introduced to the rainforest itself by having an illustration of what the rainforest might look like in particular locations. And it's nice observational activity for students because it gives them the opportunity to just sort of look and see. So here's a frog and here's a walking stick. There's different types of plants that they can see. There's a little bird in the picture. So this represents what the rainforest might look like if it's been many years since there was a hurricane. And then they can click on the 3 days after button, and now they can get a sense of what the hurricane might look like if a severe hurricane struck the rainforest. So you can see many of the trees got knocked down. Those that survived the hurricane had most of their limbs and leaves knocked to the ground. So there's a lot of debris on the ground. We now start to see different kinds of animals that are on the ground. And so those are questions that kids might have about why do we see certain things, certain species after the hurricane has struck. And then they could look again at the recovery after the hurricane. So this is 3 years later, and you can see that there's a lot of explosion of growth. There's different kinds of plants. The canopy hasn't quite closed at that point, but you can see the transition that occurs. And then since we're talking about carbon, you can overlay different cycles on top of the illustration of the environment and see a sense of how the carbon cycle actually plays out in this particular environment. And by mousing over the highlighted parts, you can then actually see a graph of how something like litterfall changes over time. And you can see that at the time of the hurricane, there's a huge spike in litterfall, obviously because all of the leaves and things fell to the ground. And then there's a huge drop-off in litterfall, and it gradually increases as the trees begin to recover from that particular hurricane. And also, as you change from one time period to the next, it actually updates the numbers and the cycle to sort of represent the changes that occurs from one time period to the next. So that just gives you kind of a little bit of a background of the Journey to El Yunque program, as well as the connection to what we're going to be talking about today. And so with that, I will turn off my screen and I will change the presentation mode over to Grizelle.
Well, I am Grizelle Gonzalez, and I'm a soil ecologist with the International Institute of Tropical Forestry, that is with the Forest Service in Puerto Rico. And today I'll be talking about litter decomposition mostly. And as you can imagine, like most people working around here in the Forest Service, I also enjoy walking in the forest, looking above ground, looking at all the trees and measuring them and ID them and seeing the impact that they have in the ecosystem. But I also like to look at the soils, deep down in the soil, because it's a system that is still seen by many as a black box. It's still very full of life, and depending where you are, if you're in the top of the mountains here in the Luquillo Experimental Forest or El Yunque National Forest, you can find these creatures, earthworms that could be up to 3 feet long, 1 meter long, that have a great impact in the soils and the whole ecosystem. So I am particularly interested in studying them and seeing what's their effects in terms of decomposition and nutrient cycling in these tropical systems, but also in other ecosystems. I have had the opportunity to look at this question in temperate as well as Arctic ecosystem. So what am I talking when I say decomposition? And here is a definition. Decomposition is a process that involves the breakdown of plant material, the transfer of organic matter and nutrients into the soil, and the release of carbon dioxide to the atmosphere. All of these things are involved in the decomposition process. So as you can see, decomposition it's very key in terms of regulating this carbon dioxide to the atmosphere. We know that carbon dioxide is a greenhouse gas which is released to the atmosphere through human activities such as deforestation or due to the burning of fossil fuels, but it can also be released to the atmosphere through natural processes such as respiration or volcanic eruptions. The regulation of the decomposition rate can be determined by 3 interacting factors. Those are the physical-chemical environment, the substrate quality, and the organisms. The physical-chemical environment, these are all the abiotic conditions like rainfall, temperature, solar radiation, the energy that gets into the system. The substrate quality here, mostly we are talking about the chemistry, whether we have a substrate with high percent of carbon, of nutrients like nitrogen, or secondary compounds like lignin or phenolic acids that make it more difficult to decay. And the third one, it's organisms. Here it's all like one big circle, but now we know that within this circle what we're referring is to the soil organisms as well as the microbes and the interaction that there is between these two. So in 1978, when describing decomposition rates across latitudinal gradients, Meyer found that the annual decomposition rate was well predicted by 2 variables, 2 out of those 3, by the annual actual evapotranspiration, which is a climatic variable, and by the lignin concentration, which is a substrate quality index. And soon after that, a lot of people went out to look at these 2 effects, how climate of actual evapotranspiration and lignin concentrations or other secondary compounds affected the decay rates for a particular study site. However, there are some limitations on this model. Today, I am just going to mention two. One is that the actual evapotranspiration rate, that is the actual water that could leave the system in terms of evapotranspiration or transpiration, it's really not an adequate predictor of mass loss rate in some biomes, particularly like in the subarctic of semi-arid regions. The second limitation is that assumes that the biota are totally predicted by climate and substrate quality characteristics, and we know that there are certain biota that have adaptations that really can cope with harsh environments or harsh substrate quality, like it is the case of the termites. But why should we care about these climate controls on decompositions in the tropics, and particularly given climate change? Well, we know that those anticipated climate changes caused by either tropical deforestation that could lead to the replacement of the vegetation from forests to grasslands or shrubland can lead to changes in the actual evapotranspiration rate, as well to the rainfall patterns, depending where you are and what climate model you use. There could be an increase or a decreased prediction in terms of precipitation in some areas in the Caribbean. And then we might expect changes in terms of decrease in the surface air temperature and changes in the cloud cover. If we look at the regional projections in terms of temperatures, using statistical downscaling, these are methods where we really look at more refined projections. For the Caribbean, we know that we are expected, the region is expected to get warm between 1 to 5 degrees Celsius, depending on where you are in the region, and also depending on the scenario that you're using. One where the A2, which is high emission scenario, which really it's high emission, but very comparable to where we are currently in terms of the emissions. It's something that we could expect for Puerto Rico around 4 degrees Celsius. So the relative control of climate and substrate quality on the decomposition process across latitudinal gradients is something that has been pondered quite extensively already in the literature, in the studies. Climate seems to be the dominant determinant of decays in areas where there is unfavorable weather conditions, while litter quality seems to be the dominant factor in sites where there are favorable conditions. So where in the tropics, usually where you have good moisture conditions, warm conditions, it might be litter quality the main determining factor. But Berg in 1998 described that if climate and site conditions are constant, then the chemical composition and also the physical structure of that organic matter can regulate the decay rate. Not many models really incorporate the organism effect. There have been some that have done so. It is not easily done because it is difficult to incorporate soil fauna effects into mathematical models. We don't know how to tease apart these independent effects of the abundance, the diversity, and the activity of the various organisms. Also, these animals are very limiting regulating factors in the ecosystem, and really they're important by far can exceed their direct role in energy release. So some of these animals are really, really tiny. You need to look at them under the microscope, and if you look at the system, the amount of CO2 that they will respire is really little, compared to really that main effect that they can have by directly increasing the surface areas of the organic or the plant material for microbes to then decompose, or the direct effect that they might have on the microbial community. The biomass of the soil fauna are not constant across biomes. In terms of biomass, the macrofauna we know are more abundant in tropical regions than they are in tundra or polar deserts. The mesofauna, these are usually divided in terms of body width or length, depending the author, but the 2 millimeter guideline here in terms of body width, more than 2 millimeters for the macrofauna, or less, then we get into the mesofauna and the microfauna. The microfauna are more abundant in temperate and tundra polar desert systems. And just some examples of the macrofauna that we see here in El Yunque National Forest. The earthworms are very abundant. We also have Homoptera, the millipedes, the beetles, ants, pseudoscorpions, spiders, and in terms of the macrofauna, that community is mostly dominated by the Collembola, the springtails, and the mites. There are different groups of mites. Within this group of animals, we have predators, we have animals that can directly feed on microbes, and these groups, the mesostigmatics and the oribatids, are the most dominant in our system. Something also that it's important to take into account of why it's difficult to incorporate animal effects into decomposition. It's because sampling protocols are really not well established or haven't been used very well in some sites and in a way that we can compare. In this slide, you see different methodologies being compared, whether you are studying animals from the litter or the soil underneath the litter you're studying, whether you are using lights or whether you're not using lights. A lot of people extracting animals from the soil and litter environment use light as a source of increase the difference in moisture, so the animals avoid the light, the desiccation, and start moving down into these funnels into the bottom where you have the alcohol or the collecting bio, where then you can have them and look at them under the microscope. The method that you use, whether you use light, the number of days that you use for that extraction will affect the abundance, the numbers of the animals that you get. And here's just a graph to show you two sites, the dry forest here in Guanica in Puerto Rico versus the wet forest in El Yunque. And we did an extraction of litter arthropods with and without light, and you see that there is a significant increase in the number of abundance that you get when you use lights versus doing an extraction without light. And if you compare the extraction efficiency in these two sites and the two methods, you see that in the wet forest, once you expose those animals to that harsh environment created by the gradient, in 24 hours you have really more than 90% extraction efficiency, and by 2 days, 48 hours in the extraction, you know, even 3, you have, you know, you're very close to 100% extraction efficiency. In the dry forest, which is the graph on the top, you see that you really need to run that extraction for 5-6 days. So you then increase that extraction efficiency to 100%. I saw somebody had typed a question. Should we entertain that question now, Steve?
Yeah, sorry, I was on mute. Yes, you can feel free to answer the questions that pop up.
Okay. Let me see. Minor question, but curious. How many samplings are represented in each condition? Since in the last figure and this one. You mean how many samples represent each of these points? I cannot remember exactly, but looking at the previous photograph, I would say more than 20 samples. And usually when you are studying animals in a system, you want to do it several times through time because they change with season. So you usually will want to do these extractions multiple times, and in each time then have enough replication so you get a good sample that you can describe the community. These animals are pretty patchy, so in some samples you might get just very few organisms. It could be 25, 30, but then there might be some samples where you get over 100 or 200. Okay, if there are any questions, please type them or come in with your voices. So I've been talking a lot about decomposition, and I wanted to spend a little bit of time, just like in the previous slides, on how I extract them from the samples. Here is a picture of the actual way where we study decomposition. Basically, these are litter bags. These are bags that can be made out of different materials, out of cloth or a plastic screening. And here you see some litter bags out of cloth, but they have different mesh sizes because I use that to play as a way to allow some animals to get into the bags and look at the effects of particular groups on the decay rates. So basically, you build these bags, and then you put an amount of material inside it. You close them, you place them in the field, all of them at the same time, and then through time, you go back and just collect a subset of them. So you start getting an idea of the percent of mass loss through time. And once you go to the site and bring the litter bag, since I am very much interested in looking at the effect of the organisms, I open the bag and then place them in this. These are called Berlise or tall grain extractors, where you take the plant material into the top of the extractor. In the bottom of it, you have there is a funnel that goes into the collecting vial where it has some 70% ethyl alcohol, and you leave that extraction to run for a week. I have these lights. These are like Christmas lights connected to a dimmer, and I don't start full blast on them. Every day I increase the intensity of that light bulb because we don't want to fright all the animals at once. We just want to create that gradient of heat and moisture and slowly allow the animals to move through the litter and then get into the collecting biome. So once you collect these litter bags, you can have a graph that looks like this, where you can plot the percent of mass remaining in the litter bag through time. That's a variable that it's very very comparable to other studies. The other thing that you can do is look at the regression, a linear regression of this relationship, and basically look at the slope of that regression. That will be the K that you see there in the equation. And that is the variable that a lot of studies has related to the decomposition compare. The greater that value, what it means is the faster the decomposition rate. And here's a graph as an example where I compare the decay rate in El Yunque, the tropical wet forest, versus the tropical dry forest, and some sites in Colorado, subalpine in the north and south facing slopes. And we can see that the animals have a significantly greater effect in the decay rate of this leaf, the Cecropia, in the tropical wet forest as compared to the other sites. The bars are green and red for the control, meaning that all the animals were getting into those bags, versus the fauna excluded treatment, where we excluded all the fauna from the sites. So you see there's a huge difference in terms of decay rates once we remove the fauna, particularly in this tropical wet forest. Another thing you could do is look at the faunal effect when you compare to the ratio of this decay rate in the plot where you have the fauna excluded versus the total. And of course, this range of values will vary widely along latitudinal gradients because of the confounding effect of the different organisms, their size, their diversity, their functionality. And in this particular site, we were able to determine that about 70% of the decay rate could be explained just by soil fauna alone. And in particular, in this system where we have, the earthworms, they are a very determinant variable in terms of decay. The animals can affect the decomposition rate directly, by fragmenting the litter, but they can also affect the decomposition rate by interacting with the microbes. And this graph basically look at the effects of the fauna exclusion of the animals on the microbial biomass in these tropical wet dry forest sites and in the subalpine sites. And we see in the subalpine sites, when you exclude the animals, you see those red bars really go up, meaning that when you exclude the animals, the microbial biomass increases. So this graph might suggest that the animals not only indirectly affecting the microbial community, but directly actually might be feeding on them, having some sort of selective inhibition on their growth.
So, Grizelle, I have a question about this graph. If you go back. So, is the subalpine, is that, you mentioned Colorado previously, so is that
Yes.
at a site in Colorado that you were studying?
Yes.
Because sometimes I get confused because there's also a forest type in El Yunque called Colorado.
No, this is actually in the state of Colorado. Yeah, temperate subalpine site.
So can you say a little bit more about why there is such a large difference in the subalpine when you exclude the microorganisms versus in the tropics?
So in this particular site, we in the subalpine, you don't have that much of the macrofauna. What you have is more of the little animals that might be actually interacting more closely with the microbes. They might be directly feeding, doing some selective feeding on the microbes, on the fungi that it's in those sites. So when you remove them, what you see is a spark on their growth, on their microbial biomass. And you know that is kind of cool. And that's why I present then this graph where the soil fauna can affect ecosystem processes, this decomposition, nutrient mineralization process through the modification of the soil and the substrate environment. The soil by, the organic and mineral mixing, the water infiltration, aeration, these are things that are more associated with the greater size organism animals, like the earthworms. Also, that substrate modification, as they pass the materials through their gut, they can affect the dispersal, the surface area of the material. But in the temperate subalpine example that I just showed, there is another way that they can also affect the ecosystem process, this decomposition process, through the selective feeding, and that's what was shown in the previous graph. I have more material that's related to hurricanes, Stephen, but I wonder if we want to get into the discussion and if the teachers want to get into more the details on how these decomposition rates can vary as related to hurricanes. I can get into that, but I also want to hear their feedback and make sure that they're with me.
Okay, so does anyone have any questions at this point for Grizelle?
I don't.
Okay, so I will show just a few more slides, Stephen, if that's okay.
Sounds good.
Okay. So another aspects of climate change, particularly in the Caribbean region, is this prediction of an increased frequency and intensity of hurricanes. As we know, the temperature in the ocean, in the Caribbean region, it's increasing, and when this temperature increase, then the frequency and the intensity of the hurricanes also increase, and this is what's being shown in this figure. 1971 to 1994, you see less hurricanes passing through the region because those are colder temperatures, but during the 53 to the 70, 95 to 2000, those were years with those were warmer years, so you see more lines and increased frequency and intensity of the hurricanes. So we know that these global models are saying that the temperature is going to be increased in terms of the projection in the next 40, 50 years. That might lead also to a high level of hurricane activity. So it's important for us to understand what the impact of these hurricanes will have in the system. Of course, we have had hurricanes in Hugo. In Puerto Rico, we had Hurricane Hugo. For example, you see this picture from the Bisley watershed in El Yunque, and then 6 months later, in the short term, there was an alteration of the ecosystem processes, and there were two main factors really that were affected: the canopy opening, where we saw greater light levels, changes in soil moisture, temperature conditions, but there was a huge increase in the pulse of organic matter going into the system in terms of litterfall and woody debris. After Hurricane Hugo, basically, what we received was about 1 kilogram per square meter on the ground, which is the amount of litter that we get through a whole year. So in a single day, in a single event, we got the amount that we will get in a full year. And the other aspect, it was a redeposition of this organic material from the canopy into the subcanopy and the forest floor. So to look at these effects more mechanistically, the Luquillo LTR, Long-Term Ecological Research program, did an experiment where we simulated a hurricane, and we tried to separate the effects of this canopy opening and the increase in the debris deposition into the system. We trimmed almost 600 trees in 30 by 30 meter plots. These were a total of 12 sites where we randomly assigned this experiment on a 2 by 2 factorial. Those were, the two treatments were trim the canopy versus not trim the canopy, and deposit the debris in the site versus not doing it. That will give us four treatments. The control, where we didn't do anything. The hurricane simulated plot, where we trimmed and added the debris. And then the two other sites where we didn't trim the side, we had the canopy, but then we added the debris. So we had this addition of debris when the canopy was not manipulated. And in some other areas, we trimmed the area and remove the debris. So in this scheme, we measure various forest responses, abiotic responses, biotic responses, and biogeochemical process. And what we did was set up a wood litter decomposition experiment. We also had a wood decomposition experiment where the plots were in the layout divided in 16 plots. We just randomly selected 5 of those 16. And we built litter bags using 2 mesh types. That is to kind of exclude the entrance of the organisms, so we could look at their effects that they will have within this context of the hurricane simulation. And we ran that experiment for 2 years. So within these 2 years, we at the initial time, we set up all the collections, and then through time we will go and collect a subset of those. And what we found was that the decay rate of these materials was significantly faster in the control plots, where we didn't affect the canopy or added affect the canopy. When we add the debris, actually that treatment, it's like a fertilization treatment, and that decay rate is accelerated. But when you trim the canopy, that was the most significantly negative effect on the system. When you trim, you see how that percent of mass remaining increases, meaning that the decay rate is slow. The T and the debris, that's trim plus debris, that's the hurricane simulated plot, and has the highest percent of mass remaining, 57%. So that was the slowest decay rate. And how do we explain these results? If we look at abiotic factors, we see that when we trim, we open the canopy, the amount of water, rainfall that goes through the forest, the throughfall, increases in the system. You see these open circles that the canopy opened. It's greater, and it takes about a year and a half for the canopy to start closing as compared to the untrimmed plot sites. And then this, What this trigger is a difference in moisture in the litter and in the soil. The litter moisture was higher when in the no-trim plots. So in the top graph, you see the litter moisture here, no trim. These are the closed dots. The litter moisture in those sites was greater than in the trim sites. And the opposite happened for the soil. Soil moisture was higher in the trim than in the non-trim plot. So this litter moisture, it's like a buffer. It dries up, but it keeps the moisture in the soil in those sites that have the canopy trim. That trigger a cascade effects in terms of the organisms. So there was a change in the microbial community, particularly in the fungi. And another scientist, Jean Lodge, was able to determine that the number of fungal connections going through these leaves was actually related to the percent mass loss. Has this fungal community change? So the animals, the mites, the Collembola, the Socophora, these animals that have mechanisms really to deal with conditions that are kind of harsh in the trim environments, they flourish. These mites, as you can see, they are like little balls with very hard exoskeleton, and they can withstand these pretty harsh desiccating conditions in the trim plot. The Collembola, you see here at the end that furcula sticking out. They can jump in and out of the places pretty easily, so that's how they can cope with that desiccation. But then there are other organisms like the Hemiptera, the Isoptoda, the Diplopoda that actually decrease in the trim versus the trim plots. So there was a change in the relative abundance of the different trophic organisms, and that was related then to the percent mass remaining, the decay rate, and the nutrients, in this particular case, the nitrogen. The total density of the animals, particularly the Collembolas, the biomass of the Collembola was significantly related to the percent of mass remaining in these litter bags. And I'm not going to get into all the details of this, but just to show the cascading effect, where in those areas where we had the canopy open, solar radiation coming in, the moisture condition in the site decreased. That changed the microbial community from macro to microfauna, and also had an effect on the organisms that you see there, from macro to microorganisms, and that relationship had a lot to do with the shredding of the material, the mobilization of the nutrients, and the litter decomposition. That later was also related to the amount of nutrients that was available for the plants to take on and then have the system recover. And that's all I have for now.
So I had a question, and then we can open it up if anyone else has questions. So the one slide that you showed of the trim versus the no trim, that you said in general, when the canopy's opened, that the decomposition rate is slower than in the conditions where the canopy's closed. And so how would you extrapolate to this idea if El Yunque is struck by rain or hurricanes more frequently? Would that sort of cause sort of a major shift in the decomposition rate for the forest as a whole, to be slower and have more debris on the ground in general?
Yes, I think here the timing of the events is going to be very important. If they are too close to each other, I would say that maybe the system has not recovered and that we don't have that much material to dump in, for example, big pieces of wood that will have lower quality. Uh, so that would be even more of a reason for the retardation of the decomposition versus if you have more leaves versus the wood. So I think it depends on the time since recovery, also on the intensity of it. The intensity meaning that maybe there could be a redistribution of this organic material from, say, slopes to valley sites, and then that also can have an effect.
So would that also have an effect on if there's a slower decomposition rate? That means that the minerals and resources are not returning to the soils at the same rate, which could affect the quality of the regrowth of the forest over time.
Correct. Yes, that is correct. And in this experiment, what we notice is that the system has kind of like check mechanisms in a way,
Yeah.
that the fungal community was able to switch, really, to change. And that also was connected to the change in the organisms, like the animals, that can also, help increase the surface areas for other microbes to start, acting in the decay and the release of these nutrients. So
And I
it's
guess
kind of neat that the animals can really serve as a balance in this situation.
and I guess we'll stay tuned because you're in the middle of doing the trim again to see what the long-term effects are of more frequent hurricanes.
Correct. This first experiment was more at the effects of the mechanisms. This main effect of, okay, whether it's the trimming or the debris deposition that have a main effect in slowing, retarding, or having an effect in the system. The second experiment is more at looking at the frequency, increased frequency of hurricane simulation. So we don't have the full factorial. Now we are mostly focused on comparing the control versus the hurricane simulated plot.
Does anyone else have any questions? Oh, while folks are formulating their question, Kalani, I'm wondering if you'd be willing to share a little bit about your experience using the Journey Tel Yunque program, and tell us a little bit about your school context as well.
Okay. Can you hear me?
Yes.
Okay. Okay. I'll start. Um, let's see. I am a middle school science teacher. I teach sixth, seventh, and eighth graders. And this past quarter, I was had the privilege of using El Yunque as a resource in my class. And one of the first things that we did when I introduced the site to the students, um, I actually printed out the resource pages with the 3 pictures, and without giving them too much information, just asked them to formulate questions that they saw, the questions that they wanted to ask as they were looking at the pictures. And we basically went through the questions, and I didn't answer any of the questions, but really just allowing the students to kind of look at the pictures like you're pointing out earlier. Here's a bird here, here's, a lizard, looking at the different types of plant life that you see from the before and the after of the hurricanes. So really just getting them to ask those questions. And then as we moved on into the koki population, I had them look at how the graph works, and you basically allowed them to play with how different things might affect the koki population, and have them to explain what their thoughts. And the last thing that we were doing was actually kind of goes back to what we're talking about today, which was on how the students, basically trying to get the students to figure out why the koki population increased. And it had, of course, to do with the amount of litter that was on the ground that allowed them to hide and reproduce. I'm sorry. So that was one of those, one of the things that we did, and the kids really loved it. They really liked reading the different articles that were on the website, and it's a tool that I'll continue to use.
All right. Well, thanks for sharing, and hopefully the presentation today gives you some ideas for using the carbon cycle component of the
Yes,
program.
definitely.
All right. Any other questions? Well, I think we're all set then. So thank you, Grizelle, for sharing your research with us about decomposition and the carbon cycle and how that is influenced and could influence global climate change.
Sure. Thank you, and good luck to all.
Okay.
Thank you.
All right. Thanks, everyone, for joining us.